intestine and respiratory epitheliums, direct life cycle, oocysts 4 to 5, very small size. When you buy a filter from Costco or anywhere else, any store, they usually do not filter these guys. They filter Giardia because Giardia cyst is about 10 micrometer. But these guys is about 4 or 5 micrometer, and they do not, cryptosporidium, they do not filter the ones that you buy from stores. Very small organisms. Microgametes and macrogametes, of course, it's an epicomplexal. They have microgametes, oocysts in soil, water, or airborne, and outbreaks in Washington, D.C. and Milwaukee. There were 2 outbreaks in the United States, and they blame cows because cows pooped in a body of water, or the sewage plant broke, and the drinking water got contaminated with the sewage water, and people became, thousands of people became infected in both outbreaks. And then, of course, they were hospitalized, and some people died. A few people died as a result in both outbreaks. Why did you do that, Glenn? Phylum Ciliophora. Let's finish this. There is not much to be said about Ciliophora. And then, so we only have Toxoplasma gondii for Thursday, and a review of the slides, and then we are done. Okay, so Ciliophora, the next phylum, mostly free living, some parasitic, some commensals, and some are thorny. Okay, and of course some are sessiles, like Suctorians. They group Suctorians. Again, your textbook classifier says it a little bit different than I do, but you know, he puts them, I think, in a different phylum. You know, but I say just larger than most protozoa. If you look at them under microscope, watch out. Under microscope, Euglena and Paramecium they do look alike, but Euglena is much smaller, and Paramecium is much bigger. Okay, so look out for them. Of course, in Paramecium you should see cilia, you should see huge nucleus, and other. Multinucleated, they have a macronucleus and micronucleus. Macronucleus is used for metabolism, something you learn in Bio 1 to make protein molecules and so on and so forth. Micronucleus is used for reproduction, cellular reproduction. So they have 2 nuclei, the macro, the larger one, and micronucleus, the smaller one. Undulating membrane cilia are fused into a sheet, and cytostome is the mouth of the animal, oral groove. These are structures. We have it on the model. 2 contractile-vacuole system. Already talked about that. Passage of water and fluid in Paramecium. Do not worry about this so much. When I took zoology a long time ago, I had to memorize that, but I don't think it's that critical for our purposes in here. You're learning a bunch of other stuff. Okay, so I'm not worried about that, but that's pretty much the cytoproc. You should know it on the model. On the model, we have cytoproc or cytopage, 2 names for the same thing. Cytopage, cytoproc. That's where the food material gets out. Reproduction, binary fission, micronucleus divide mitotically, and macronucleus elongates and divide amitotically. A means without mitosis. Right? Conjugation, and when 2 Paramecium come together and exchange nuclear material. And 2 Paramecium. Today I will put 2 microscopes there, one with a binary fission and one with conjugation. Make sure you look at it. You will not see it there next time. So it is in the pictures or in your lab manual, your textbook. Autogamy means self-fertilization, example of Balantidium coli. And I will talk about Balantidium coli here. So these are the structures on the models that we have. I'm hoping you would know. So you have the contractile vacuole, right? I talked about that a long time ago. And these are ampulla around it. The ampulla are connected to endoplasmic reticulum, right? I hope I'm making some sense. Macronucleus, they forgot to label micronucleus. Okay, then you have the oral groove right here, cilia, of course, and then oral groove, it comes to cytostome. They forgot to mention here cytopharynx and cytostome, and then food vacuoles, and then food vacuoles after being the waste material gets out through cytopage or cytoproc, and in bio 1, you learn what happens with lysosome and food vacuoles and what happens, so on and so forth. We're skipping that in this class, right? You learn that in bio 1. Here are some examples of the ciliophora. Here again, another picture of it, and then another name for oral groove is gullet. And then there is one other thing I think I asked today. This is a term I would like you to know, is trichocytes. At the base of the cilia, at the base, they have these structures called trichocytes. One theory is that they hold the cilia in place. Okay, but anyhow, again, contractile vacuole, one is being full of water, one just excreted its water out. Do you remember I talked about that? Okay, this is my drawing, but I should have deleted it. I deleted that from your notes. But here, the same. This one says the same thing. Conjugation. Let's go over conjugation. So two paramecium come together, and then meiosis 1. Do you remember in biology 1? You learn meiosis 1 has 2 stages: meiosis 1, meiosis 2. Macronucleus there. Micronucleus undergo meiosis 1. Micronucleus undergo meiosis 2. And then what happens? The macronucleus disappears, the micronuclei disappear, all of them except one. I draw it in star shape. And then these undergo mitosis. The blue micronucleus and macronucleus disappear, right? Am I making some sense? Macronucleus disappeared, macronucleus disappears. And then these blue micronucleus, after gone 2 stages of meiosis, undergo mitosis. Okay, on the separate one. Then they exchange. There is one that is stationary, and there is one that is mobile. So this one, for example, moves to that one, and that one moves to this one. Now the 2 micronuclei are fused together, right here. So you have DNA material from this one in that one, and from DNA material from this one in that one. Macronucleus reappears again, and then they separate. The 2 paramecium separates, and then each one of them undergo binary fission. When they undergo binary fission, the nuclear material is same as these. This one goes under binary fission. After going through conjugation, the nuclear material is different than the original one. And what is the advantage of that? Why? You studied this in Bio 1, but I'd like to ask again, what is the advantage of sexual reproduction? Or what is the disadvantage of asexual reproduction? Nicole? Huh? Anybody? Nobody? Come on, every semester somebody give me the answer.
Isn't it diversity?
What diversity?
For better natural selection.
Natural selection, diversity, you're not hitting the target. But yeah, the genetic makeup is going to be different, right? Of these guys, the genetic makeup of this one is different than that. So if a disease or a natural destruction, you say natural selection, let's say a natural destruction comes, what will happen? This one might destroy, it might kill that one, the natural disaster, but this one will survive. Why? Because this has different genetic makeup. It can handle that disaster. Okay, that's why we are all genetically different, right? Except identical twins, even though there are some differences there too, but they're pretty much the same. Okay. Here's a list that you know more than this. Is that right? You all know what I'm talking about? You already know more than this. This is just a short list. This is not all, you guys. This is not all. Elizabeth, am I, or I'm not? Yes, you are, Amir. Very good. Okay, do not, well, yes, flagellates, yes, Trypanosoma cruzi, I already mentioned that. Answering Glenn's question, indirect life cycle, it needs a Trypanosoma means a Chagas disease via blood-sucking bugs. Okay. Trypanosoma brucei gambiense, Trypanosoma brucei rhodensiense, Leishmania, you already know those. Giardia, direct life cycle. These are all indirect life cycle. Trichomonas vaginalis, yes, we talked about that. Again, direct life cycle. Amoeba, entamoeba histolytica, direct life cycle. And then you guys should know the mode of transmission. And then the pathology. What kind of disease do they cause, right? You do have this in your PowerPoints, right? You should know these. And then, of course, adult parasites, location, host. Yes, you already know most of these. And then he also gave you where in the world they are located. Widespread outside of Europe and North America. But they're found all over the world. Cosmopolitan, it means all over the world. That's what the term means. Cosmopolitan, all over. And then he mentioned American and Europe, of course, widely distributed outside of North America and Europe. So I already mentioned the locations. Sporozoans, again, that is old term. You don't use that term anymore. But Plasmodium, again, the location, everything else. Toxoplasma gondii, I will go over it on Thursday. A little bit more detailed than what your textbook gave you. And then, of course, ciliated Balantidium chloride. And I believe all of this, except entamoeba histolytica, except that one, we have them all in the lab. These two, you don't have to tell them apart. Trypanosoma cruzi and Trypanosoma brucei, you don't have to tell them apart. Am I done? Okay. Then